Hello everyone, my name is Beatiz Estevam, and I will present the work about the identification of potentially new CAZymes acting on mannan and xylan polysaccharides to the ISMB 2022. This work was done with Danilo Rocha and Diego Riano at the Laboratory of Computational Evolutionary and Systems Biology at USP. Firstly, despite the relevance of using renewable energy from biomass, especially in a scenario of growing energy demand and environmental concerns, an important fraction of the energy stored in plants is hardly accessible for human activities, requiring the usage of pretreatments to release the energy stored, like using carbohydrate-degrading enzymes as some CAZymes. And two important hemicellulosic polysaccharides in monocotones are mannan and xylan, and there is interest in finding novel CAZymes that can break them into mono or oligosaccharides. Then, as a consequence of the continuously decreasing costs of genome sequencing, there is a large number of genomes deposited in public repositories. However, exploiting protein characterization and structure is costly and time spent. Based on this, we sought to identify CAZymes by performing phylogenetic analysis and selecting sequences from less studied clades that have potential to break mannan or xylan to target proteins that have a greater potential among the various sequences available. To reach this, firstly, we made the prediction of CAZymes from archaea, fungi, and bacteria groups' genomes deposited on an NCBI database. Simultaneously with this protein sequence acquisition, we identified what CAZymes have a record of mannan or xylan degradation, and these ones we consider as families of interest. And for each family of interest, we extracted structure and characterized tabs of CAZymes database using web scraping to build and work with our personal CAZymes database. From these two actions, we've selected 15 families of interest for mannan and 22 for xylan. And we've obtained more than 1.3 million predicted proteins for in the genomes for mannan, and only 2.7 thousand studied protein sequences also for mannan. And for xylan, we have more than 1 million of predicted sequences, and almost the same amount as mannan for the exploited sequences. All this from approximately 380,000 assembly accessions, finding more than one enzyme predicted per genome in all the families of interest. And once we have the protein sequences, its information, in mannan and xylan's families of interest, we selected the sequences that belong to the families of interest and analyzed its status. And by its status, I mean if the sequence was predicted, a predicted sequence, a sequence with structure or characterized informations. For mannan, we have three major families in sequence amount. They are GH1, 2, and 3. Adding to this GH5 and 26, the five top families in sequence amount represent 84% of the total amount of CAZymes sequences in mannan families of interest. For xylan, the distribution is more even, with the five tops representing 73%, but some families such as C1, C4, GH31, and GH43 still overrepresented in sequence amount terms. For mannan and for xylan, the group that has more CAZymes per assembly accession is bacteria, with some exceptions, which fungi takes that position, such as GH47 and GH134 for mannan, and CE16 for xylan. We also have cases where bacteria and fungi groups are similar in the contribution to the CAZymes sequence amount, situations seen for GH11 and 62 also for xylan. After this, we made a clustering by sequence similarity, and we aim to build phylogenies to finally identify poorly explored clades and clusters. From xylan, generated clades for xylan, we saw that all families have more than 50% of the clades not studied. That is, the more than 50% of the clades for xylan doesn't have any sequence, any studied sequences even for function and structure. And the same scenario was found for mannan, except for GH134, that has 40% of the clades not studied. For this family of interest, we have 508 predicted sequences, 5 and 7 sequences with structure and function informations respectively. As a discussion, we bring the small amount of characterized and structure exploited enzymes. Data seem especially in the number of not studied clusters, and that stands for an indication that there are still many morphotypes that have not been exploited yet, and because of that, identifying only the clusters not studied may not be enough to precisely highlight potential enzymes to degrade mannan and xylan, needing a method to rank clusters based on their connection to the degradation of these carbohydrates. Another interesting point is the almost complete absence of overlap of the CAZymes families that degrade these two different carbohydrates, the mannan and the xylan, indicating that mannan and xylan bond catalysis is considerably different. To conclude, our next steps involves finish the phylogenies and identify the less studied clades, identify bond catalysis related to mannan or xylan using the protein ZC code for studied sequences, rank the clusters based on the quantity of studied proteins and mannan or xylan bond catalysis quantity present in these clusters, and lastly, explore some proteins 3D models. Thank you for your attention.